Hello, my name is Rojin Aflaki and I'm a student at Holland College Post-Certificate Program. And I'm here to present my poster and let you know about my individual research project. So the topic of this project was effect of chemical treatment using copper disulfate and L-Cysteine on melanogenesis and melanin antioxidant activity in Bacillus licheniformis. So it is about a bacteria called B. licheniformis. This bacteria has a specific characteristic that makes it suitable and interesting for this experiment. It can produce melanin, one of the types of the melanin that is also in our own skin. We have two types of melanins in our skin, eumelanin and pheomelanin. And eumelanin has higher antioxidant activities than pheomelanin. And this bacteria can produce the eumelanin. So in this experiment, I'm going to treat my bacterial cultures, with two different chemicals. One of them is L-Cysteine. L-Cysteine is an amino acid, and in the presence of L-Cysteine, because it has a thiol and has a sulfur in its structure, the monomers of the melanin would change subtype to pheomelanins, and it would cause them to have less antioxidant effects as already described. The other chemical that I'm going to treat it with is copper disulfate, because copper is actually a precursor for creation of the tyrosinase enzyme. What is tyrosinase? Tyrosinase is the enzyme that causes the melanogenesis in both bacteria like B. licheniformis and in human body. So it is hypothesized that the treatment with copper disulfate would cause higher antioxidant activities, and treatment with L-Cysteine would cause less antioxidant activities in the culture. And the objectives of this experiment are to first produce the melanin from the B. licheniformis culture, and then it is to extract the tyrosinase enzyme and test the enzyme assay on the tyrosinase, and after that it would be to extract the melanin and try TLC, optical density readings, and antioxidant assays such as DPPH and Falling-Kalkal. So the methods for this would be like the bacteria would first be pelleted, and the growth curve and the melanogenesis rate would be measured. The cultures would be treated with the chemical treatments, different doses of copper disulfate separately, and also different doses of L-Cysteine separately. So after those treatments are done, after 72 hours of time when the melanin is produced, the melanin and the tyrosinase enzyme are extracted from the cultures, and on the tyrosinase enzymes, the BCA assay, the SCS page, and the enzyme assay were performed, and on the melanin, the optical density reading, the TLC, and the DPPH and Falling-Kalkal were performed. As a result, we can see in 4 different cultures that we had, which was 50 degrees centigrade and 37 degrees centigrade, both constant and refreshed. The only one that could actually provide melanin was the 50 degrees centigrade culture that had its media refreshed daily. Another thing that was observed in this experiment was the optical density of the melanin, and it was observed that the density of the melanin had a direct relation with the copper disulfate concentration, and it was a significant amount. But for the L-Cysteine, there was not a significant change in the optical density reading. On the other part, we also did the enzyme extraction, and the enzyme assay was performed on it. The chemical treatments didn't have any significant effects on the tyrosinase enzyme assay or enzyme activity. In Falling-Kalkal, we didn't see any significant changes in L-Cysteine treatment, but we saw a significant increase in the copper disulfate treated cultures. On the other hand, in DPPH, we didn't see a significant change in the copper disulfate treated cultures, but we saw a significant decrease in the L-Cysteine treated cultures. So B. licheniformis actually could produce melanin. We could extract melanin, we could extract the tyrosinase, and the enzyme activity was performed. So based on the enzyme activity, there wasn't any significant changes based on the chemical treatments on the enzyme activity. But about the DPPH part and the FNC part, unfortunately, we can't be so certain about that part, because Both copper disulfate and both L-Cysteine could also interfere with those antioxidant assays. And for a better understanding of the antioxidant assays, it is essential to have further studies with better and more normal controls. And last but not least, I would like to really thank and appreciate the opportunity I was given to have this opportunity to learn all this sweet stuff and give myself a chance to learn under the kind supervision of Mike, Jesse, with sweet help of Marla and Crystal and Shonda. Thank you so much, everyone.